All right. So let's see. Today's Monday. So tomorrow in lab, you'll be doing a survey of the bryophytes and the seedless vascular plants, which I will shift into very quickly here. And then also check on the growth of your algae that you put in those test tubes with different nitrogen and phosphorus levels. And we'll take a peek at those tomorrow in lab as well. All right. I think we left off here on Friday looking at sushi. People like sushi. I see a lot of nods. I try to like sushi so far. Any favorites I should try? Is there a particular recipe, like at tamaki and tea or something that someone's tried? Nothing in particular? Yeah, I know. Maybe if it had enough mushrooms in it. Is there one with mushrooms in it? That I could probably do, especially shiitakes or something like that. Anyways, so I'm still trying to like it. So I modified the slide set and reposted it because I found a couple of things. So this was something I just saw on a website that is looking at all things algae in terms of commercial production. And so this is one of their news blurbs that they had posted where they're looking at essentially using seaweed material. And I mean, I've already known how seaweed can be added into soil as a good soil amendment. I've known about that for a long time, but this idea of the plastic, creating plastic out of seaweed essentially. And so, seaweeds have a lot of, polysaccharides that are part of their cell wall structure and all of that. And they think that it can be used to replace the polymer type plastic. So we'll see. So that could be another way to try to fight the issue that we have especially in our marine systems with just so much plastic. If we could find a way to break it down, so they found bacteria and fungi that can do that, and then if we can replace it with something biodegradable, that would be a nice 1-2 punch. So we'll see what happens. Diatoms, we talked a little bit, and you saw that in lab, how last week we got to look at diatomaceous earth, and we saw water from a local river containing all kinds of diatoms. So a couple of things about diatoms. When they are also being looked at for human nutrition, you can extract nutritious oils and fats from them for human nutrition. So that has been going on for a while now. They're looking at them for diesel fuel, extracting the oils that they produce for diesel fuel production. They have silica in their cell wall. And so that has been taken advantage of for a lot of different things. So essentially the dead bodies of diatoms, they don't break down because of the silica. And so when they're dead, they call them diatomaceous earth. And so we had a little tiny bottle of that in the lab that you could sprinkle onto a microscope slide and look at them under the microscope. But the dead bodies, essentially, of the diatoms, they use them for all kinds of things, filtering material. So like in aquaria, swimming pools, all of that, you can use filters made from diatoms. Toothpaste, you might look on your toothpaste ingredient list, see if any diatomaceous earth might have been added in. Some of the more natural, so-called natural toothpaste often have that. They put it on reflective paint on the highways. So, when the white lines on the highway really shine up brightly when you have headlights on, a lot of those are due to the reflective paint. And then adding them into soil, like we use diatomaceous earth in the greenhouse when we have pests that are, the creepy crawly pests that spend some time in the soil to either pester the roots, or they sleep in the soil and then come up onto the plant and chew the plant. If you put diatomaceous earth into the soil, it's sharp. And so a soft-bodied insect has a hard time getting around all that those sharp shards of glass, more or less. And so that can actually help prevent pests from getting on your plants. So, you know, if you don't want to use nasty insecticide, this is a really nice strategy. Oh, the other thing too, I was reading more about using diatomaceous earth for pest control. They say not only is it sharp, but it also kind of sucks the moisture out of the insects. That I didn't know. I wasn't privy to that information until I started reading more deeply into it. So it actually kind of dehydrates the insect as well as cuts them up when they're crawling over the silica. So this is a flow chart taken from a published paper from a couple of years ago, where the scientists were trying to demonstrate the many and varied uses of diatoms and the potential for diatom use in commercial application. And so this is all, you know, things that are relevant to today's issues and concerns that we have. And so again, you know, the diatoms are part of the kingdom Protista, mixed in with the rest of the algae, and so they're being heavily looked at because again they're very small organisms that are easy to culture in lab environments. You don't need a ton of space, and the ability to extract really useful things from organisms that can grow in a small amount of space is very attractive to people in the business world. Phytoremediation, that word, by the way, if you've not seen that word, phytoremediation here, that is, anytime you see PHYTO in a word, that means plant or something plant-like. And so phytoremediation, what they're saying here is you can use plants to remediate toxins in the environment. And so that's a huge field of research. There's businesses. One of my former grad students, MC, when she got her master's from here, she went and worked for a company in Chicago where they were looking at phytoremediation, bioremediation. And she really liked that job because she got to fly around all around the country and go to these different sites where there were issues that, so she would go to the actual contaminated site and she'd have to do an on-site assessment and then bring samples, take samples, bring them back to the lab, and then they would run all their tests, and then they would come up with like a plan of action that they would then she'd fly back to that site and work with the people on how to remediate that contaminant. She really liked that job. She liked to travel, and she liked the idea of helping clean up some of the nasty toxins that we polluted our environment with. So, let's see. And then this is from, I think, the same article that was published, and I just wanted to put this on here again to get you thinking about all the different useful products and services that they're finding with diatom use and some of the different companies, where they're located. So this isn't just something in the United States, this is something globally, as many of these businesses looking at the use of algae is global. There's many different companies worldwide looking at this. So something to consider and think about, and just you already know that they can be quite beautiful to look at, living or dead. They are very beautiful. And then if you want to go check them out, Madison, Wisconsin is having a conference October 9th, 10th and 11th, right on the university. I think it's at the, oh, I'm blanking on the name of the, oh yeah, it's Monona Terrace. It's right on the lake, Lake Monona. Monona, is it? Okay. Yeah, it's right on the lake there. It's very nice. If you're over 21, you can get a beer. They have a beer garden there. So, but if you've not really thought about conferences before, you may want to think about that, in the sense that going to conferences, and you don't necessarily have to go there to present, but just going there and going to sessions and networking, talking to people, can be a great way to get your foot in the door at some of these companies, because they go there. A lot of companies go to these conferences and then they are looking to hire, and they'll have like job announcements posted right on site. They do interviews right on site. And so it's a really nice way to start to network So this one is only, what, 5 hours away, not too far. So if you have any interest, you may want to consider that idea. Okay. All right, so I am going to shift gears. So we've covered the fungi, algae to the extent that I can. And then the next topic, so now we're going to continue with looking at other phyla and getting into, terrestrial plants now. And so the algae primarily are water-dwelling, ocean-dwelling, some freshwater, although there's certainly some that live on land. But now we're going to shift into the plants that are very common on the land. So the bryophytes will be the first one. So with the bryophytes, what I'm going to do, I think I've posted in EduCat, I think both the bryophytes and the seedless vascular plant PowerPoints are posted. I'm going to say a few things about the bryophyte slide set, and then I'm going to shift into seedless so that you have the necessary information before you walk in the lab tomorrow. And then on Wednesday and Friday, I'll finish up these 2 slide sets. But I'm going to jump around a little bit, so you might want to pull up the seedless vascular plant one as well. Okay, so the bryophytes. Bryophytes are the cutest little plants. So we're looking at mosses, liverworts, and hornworts. Those are the 3 groups of plants that fit into the bryophyte category, all right? And so these are plants that are sadly often overlooked because they're so petite. And they, in this area of the country, they grow mostly on the ground. And so it's easy to overlook them. And if you can go into other areas where they're more epiphytic, growing on branches, they're a little bit more noticeable because they're up higher and we can view them a little easier with our when we're out hiking around. But what we're looking at here, here's just 3 pictures. The mosses are up top. The liverwort, an example, is in the middle, and then a hornwort on the bottom. So really petite plants. They are petite for a reason, all right? So when we look at bryophytes and say, what makes a bryophyte a bryophyte? Most of them prefer moist habitat, but surprisingly, some bryophytes, especially mosses, can live in really dry environments for long stretches of time. So there are some kind of outliers that are amazing at handling some more extreme conditions, but in general, most prefer moist habitat for most of their life. They're small and petite, and that is mainly because they do not have true roots or leaves. So they have no vascular system. They have no way to take up water from the soil through roots. And so they have to get hydration and mineral access a different way. And the way they do that is the rhizoids that are, it sounds like a root word. It sounds like it's something to do with roots. And it kind of is in the sense that they're just extended thin cells that can help anchor the plant. All right. But the rhizoids don't have true vascular. So they don't have true xylem and phloem. They don't have that. So if they don't have any xylem or phloem, that means that when they need water and minerals, they have to just get that from the environment, from rain and from dust, from fog. All right, so they're reliant on that kind of humid environment, rain events frequent enough to keep them hydrated and get them their mineral nutrition. Of course, they can do photosynthesis, so they're making their own sugars, but plants still need minerals. And so this water and mineral idea, it's just coming from their environment. So rain and dust, usually snow doesn't do it much for them because they're pretty dormant, except on really warm winter days, they might activate for a little bit, but around here, most of the time they're dormant in the winter. So because of this idea that they don't really have a true vascular system, they have to stay small. And the trade-off is they don't grow really quickly either, with one notable exception. Segment peat moss, when you compare them head to head with other bryophytes, they grow quickly. but they still grow fairly slowly if you compare them to angiosperms, for example, right? So sphagnum peat moss, if you buy peat moss at a gardening store for your potted plants, that's what we're talking about. That's just dried peat moss that is being used in the soil, the potting soil that you buy. One other noted thing is that they need to have free water in their environment in order for sexual reproduction to occur, because the sperm have flagella and they have to swim to get to the egg. If it's too dry, they don't make it. So we say they need free water from the environment, right? So in general, bryophytes are really petite. They just they're at the whim and mercy of their environment in the sense of how they get their water. They can't get it from deep in the soil. They just have to get it from the rain and fog and mist, dust. So the mosses, I want to talk a little bit about. They're the largest group of bryophytes, and they're probably the one you're most familiar with. So I wanted to start with them. However, a big point to make is that I'm going to give you a list of things that are not mosses, and I'm so mad that they named these other plants with the word moss in it because it gets so confusing. So please be aware. Irish moss, we just worked with that last week. Is it a moss? That's the one that you boiled to get carrageenan. That was a red algae. So unfortunately, they called it Irish moss because it's red in color, and actually along the Irish coast, Ireland coast, you can find a lot of it. But that's algae. Reindeer moss. Does anyone know what reindeer moss is? Lichen, they're a wonderful plant, but they're not mosses. Lichens are totally different beasts. They're fungi and algae, sometimes cyanobacteria mixed into it. Club mosses, that's the next group of plants we'll talk about. Club mosses are actually a type of seedless vascular plant, so they're way not a moss of any kind. And then Spanish moss, if you're from the southern states, Spanish moss you might be quite familiar with, and that's actually an angiosperm that's in the pineapple family. So that's definitely not a moss. So Spanish moss are the epiphytic plants that I like to hang off or dangle off of branches in some of the southern trees. I love the look of it, but then I was talking to someone who said, "Oh no, it's awful," and I said, "Well, you don't like the looks of it?" And she said, "No, not that. It's if you get a lot of it in your yard or driveway or whatever. I guess there's a lot of insects that hang out in it, and it can get a little creepy crawly, and probably just having the plants blown off the tree and in your way, annoying, I would imagine. So, but anyways, so those are not mosses. So please make sure that you get that straight in a botany class. These are not true mosses. All right, so the bryophytes. Here's a phylogenetic tree, a very shorthand version of one, just kind of giving you a broad picture of how things are related to each other. And the last, so here you can see at the bottom of the tree is the ancestor, the green algae, all right, of the rest of the plants. And so you can see quickly coming off the base of this tree, you follow that up to the liverworts. So the closer it is to the base, the more primitive those plants are. Okay, so the liverworts are pretty primitive. Then you see the hornworts are next coming off that tree base, and then you see the mosses. Okay, so those are the three that we're focusing on. Those are the bryophytes, and then the rest of the tree is the other groups that we'll eventually get to. Okay. And then they're also mapping out a few distinct phyla, but we're not going to get into that. Okay, so when you look at mosses and you're out hiking around, try to pinpoint these two different morphologies. The two big categories are cushiony and feathery. That's how they describe them. So cushiony means that they're more erect and upright, and growing kind of straight up off the substrate, whether it's a rock or soil or whatever. Feathery, and that's a nice image of feathery. It looks almost like fern fronds, maybe, or something like that. And so those are the two main morphologies, how they look to the naked eye. And then here's another picture of, so this is cushiony, where it's growing up off the substrate, and here's one that's more feathery. Okay. So they're very, around here anyway, most mosses are on the ground or on cliff faces, on rocks. And you may see, depending on what part of the summer you look, spring, summer, fall, you may see like two parts of the body. You'll see the greenish part that you normally think of when you say the word moss. And then you see this other attached part that often will be growing up off the other part of the moss body. So you're going to learn about this in lab, and we just are going to briefly talk about it and mention it, and then when we get into the answer, we'll talk more. But what you're seeing is, we call it the alternation of generations of the life cycle. Alternation of generations. So you're going to hear more about this when we get into the angiosperms. But basically, when you look at terrestrial plants, they have like two phases of their life that they exist in, and sometimes you see both together. And so with the bryophytes, all of the terrestrial plants have these. The gametophyte phase, the gametophyte. Again, P like you know, something to do with plants. Gameto is referring to gametes. Gametes are the sex cells. So we're thinking about sperm and egg. Okay. So gametophytes, their job, when they're in the gametophyte phase, their job is to make gametes, which are sperm and egg. Then the other phase of the life cycle is called the sporophyte. Sporophyte phase. Sporo is referring to spores. Okay, so their job is to make spores when they're in this phase of their life. Okay. So here on this picture, what you're seeing, all the greenish part, that's the gametophyte body. Okay. And then this stalk that's sticking up off of it with the green cap to it on the tip there, that's the sporophyte. And the sporophyte, it's like firmly attached to the gametophyte, like they're you know, physically attached to each other. With the bryophytes, they do this. Okay. And so when they've completed sexual reproduction, you're going to see this sporophyte phase growing up off like this. In this case, it grows up straight. And then you see this cap. What that is the sporangium. All right. And I do want you to know that word. So the sporangium is that structure at the very tip of the stalk. It looks green here. Okay. And if you were to dissect that open and look inside, you would see a collection of spores inside. Right. So that's where they actually have the spores. So this sporophyte won't show up unless there's been sexual reproduction. Okay, so if you're walking around and you see mosses and all you see is the green gametophyte body, you don't see any sporophyte, you know that they haven't gone through sexual reproduction. But if they have that sporophyte sticking up, in this case off the gametophyte, then yes, because those spores are the result of sexual reproduction. Then eventually what happens is over time that sporangium will start to dry out and turn color. So it starts out green and it kind of goes light green, then eventually brown and tan. And then that will crack open, split open, and release the spores. And the spores will, you know, one of the reasons why they're up off the body of the gametophytes to spread the spores in the air current, get them up a little bit so they can spread by the air, right? So if you see both the gametophyte and sporophyte phase, you know they've gone through sexual reproduction. And the last thing that they do to complete all of that is they want to spread those spores out to conquer new habitat. So eventually those spores will get released. Are there questions on this idea? So you'll hear about this a little bit more in lab. And I don't know if we have mosses. It's hard at this time of the year to find mosses that have both the sporophyte and the bryophyte, but we'll see. But mosses are quite diverse in the different species and how they look. They're really quite beautiful. Last couple of plant sales that we've had, the greenhouse has plant sales every year. And the last couple ones that we had, we made terraria with mosses, and that was really successful. A lot of students really like that. They're cute little glass terraria with mosses in them. So really pretty, nice, lovely green thing to have around when it's the dead of winter. This is the Olympic National Park. If anyone's been there, has anyone been there? So that's the west coast, Washington State. And it's in the Olympic Peninsula area, I guess. I've never been. I got to get out there though, because I've seen pictures. My good friend went there, and when she was showing me pictures, it was just so intensely green because there's so much moss there. It's a perfect condition for the mosses to grow. It's a temperate forest. It's the only temperate rainforest that we have in the continental United States. It's the only one. It's not tropical because it's far north, but it's a rainforest. Another image. So you can see here how on the branches they're very obvious when they're up on the branches. But around here, when you're out hiking around, it's very easy to miss them. Here's I did not take this picture, but really good picture of water droplets off the sporophyte and the sporangium tip. Okay. So mosses are probably what you're most familiar with. I'm going to jump around a little bit now. I want to show you the liverworts. Liverworts you're going to look at as well. Hopefully we have some living in the soil environment. So again, when you look at liverworts, there's two main body types. Leafy is one of them. And actually most liverworts are leafy. And then there's the thallus type. And so even though the leafy are more numerous, often when we teach about liverworts, we often show you thallus liverworts. So it's a little confusing. So, but just be aware, leafy liverworts win. There's more leafy liverworts out there. The thallus ones have some additional structures that are cool to look at. So that's why they're often the one that we focus on in botany classes. But the leafy, well, both leafy and thallus, they can grow in really nice wet areas that mosses sometimes they get too wet, but the liverworts can handle quite well. And that I'll show you some pictures of both of these. The thallus one looks more like a leaf, like an aspen leaf. It's bigger and broader than the leafy liverworts. Leafy liverworts are more delicate looking. I'll show you some pictures. But the common liverwort that we focus on in botany classes is the Marchantia genus because it has some interesting other structures to look at. And so I'll show you some splash cups. They're called gemma cups, splash cups. And it's their way of going through asexual reproduction. And so these splash cups contain little tiny pieces of tissue that break free with a raindrop and splash out. And that's their way of distributing themselves into new territory nearby. So leafy and thallus are the two main body morphologies. So here's leafy and thallus. So you can see the leafy is much more delicate looking. The leaves are very small, overlap each other. Rib, really small. They're not true leaves. Often refer to them as leaflets. And then the thallus is more flattened, more broad design. So this is Marchantia genus. You're looking down on thallus, liverwort, and then you see those cups. So those are the splash cups that hopefully we'll see if we have any showing up on the Marchantia that we have. Then if you look closely, you can see on this, there's some pieces of tissue. If you look really carefully, here's another one. You can see little bits of tissue inside those splash cups. So if you can imagine a drop of water hitting in there just right, it'll splash those pieces of tissue out. And it's just their way of kind of cloning themselves and then splashing out into new territory. Okay, so I think we also have in the lab some aquatic liverworts that maybe you might be familiar with if you do aquaria. Have anything in aquaria. Riccia is a aquatic liverwort, and they're trying to show all the oxygen being produced there, all the bubbles. And so that's why people add it into their aquaria. They're trying to enrich the aquaria with oxygen. You can certainly use a bubbler, but some people like to add in the living Riccia for the extra oomph of oxygen. Okay, so mosses and liverworts. We'll take a peek at those, and then I wanted to show you hornworts. So hornworts aren't super common. There's not very many. So far, they've only isolated or discovered 100. And Amphoceras is one that can be found around here in more temperate regions of the world. And they call it hornwort because it just has this big long spike that kind of looks like a horn. And that big long horn or spike, that's where the spores are located. So it's kind of different. And that whole long spike will split open and there'll be spores that just kind of spill out of there into new territory. So they're very petite and small, and you can see they kind of look like liverwort. You need to look at like Chincha. And here's an actual picture of it in its native habitat. So very petite, really easy to overlook. If you see it in this area, it's a good find because they're kind of hard to find. Okay, so bryophytes, that's pretty much it in terms of their like basic biology. We have the mosses, the liverworts, and the hornworts. And then there are some other things I do want to mention about them in terms of their applied uses, but I want to shift now over to the seedless vascular plants so that you can get a basic introduction of them as well before lab. Okay, so I'm going to switch into the seedless vascular plants. And as you're studying this, I really encourage you to think about the bryophytes and then think about the seedless vascular plants and kind of do like a compare and contrast, like what's similar and what's different about them, because that helps you see their evolutionary progress of these different groups of plants. And so the name, this group is called the seedless vascular plants. So they don't have seeds, but they do have vascular tissue, right? So that's very big improvement in some ways compared to with the bryophytes who do not have seedless vascular plants or vascular tissue. So what we're looking at here there's basically 4 groups of plants that fit into this category. So the top image is a club moss. So we'll look at club mosses. We have those a lot around here. The top right image is a whisk fern. Those are very hard to find and they're not around this area. The lower left, that's called horsetail. We have a lot of that around here, and right in the city as well. And then the lower right are ferns. Okay. So when you look at the seedless vascular plants, here they're trying to show like I like how this image on the left, they're showing, the mosses, liverworts, and hornworts. They're non-vascular. They do not have vascular tissue. And then you see this shift where all the rest of the plants after that have vascular tissue. So the seedless vascular plants are the most primitive type of vascular plants. Okay, and then you have the gymnosperms and angiosperms. They're vascular too. And then this one on the right is also showing you a similar process here, where you have the bryophytes, colonizing the terrestrial land first, and then through the evolutionary process, you see groups of plants that start to innovate and evolve vascular tissue. And then if you go back to that phylogenetic tree we looked at in the other slide set, so what we're looking at are this next series of lines here. So the Lycophyta, Psilotophyta, Sphenophyta, and actually they've renamed the fern. It's no longer Pterophyta. It's a different one. But it's these 4 lines now that we're focusing on. So these are the earliest, most primitive vascular plants. Okay. So the common names: horsetail, whisk ferns, club mosses, and true ferns. And that idea of having vascular tissue, their ability to have true xylem and phloem, what that allowed then is that they can get into much drier habitats than the bryophytes. So they can get into even into desert conditions. Okay, but they can get into drier habitats compared to the bryophytes, and can be very successful. So they can really adapt and thrive to drier habitats. So they have true xylem and phloem, but they're still primitive. So they have the more primitive version of xylem, which are tracheids. Tracheids are more primitive. Do you remember there's 2 forms of xylem? The tracheids. What's the other group or the other type? Starts with a V. Tracheids and vessels. Yeah. So the vessels are more advanced. You see those in the angiosperm. Phloem cells are, when you look at primitive phloem, it's sieve cells. And then the more advanced phloem are the sieve tube elements or the sieve tube members, depending on who's saying it. Okay, so that's very, very different from the bryophytes, the fact that they have vascular tissue. However, they both have to have free water for fertilization because they have to get the sperm over to the egg, and you need free water for that to occur. So that's similar to what we already saw with the bryophytes. They have, similar to the bryophytes, they have chlorophyll A and B, and they have carotenoids. And when they do photosynthesis, if they have extra sugars, they'll store it up as starch. Okay, so some of these last 3 bullet points are similar to what we see with the bryophytes. So one thing we're going to try to look for tomorrow in lab with some of the seedless vascular plants is that there's a new structure to introduce called a strobili. Well, strobili is plural. Strobilus is singular. Okay, so the strobili, it's something that you can visually see with the naked eye, and it's distinctive. So you'll be able to point it out once you learn about it. So what it is it is a bunch of sporangia, which means that we know that sporangium is singular, sporangia is plural. That's the area where there's going to be some spores enclosed in some kind of container. I usually think of it as a sack. It's a sack of spores. And if you put a bunch of those tightly together in one portion of the plant body, it looks distinct, and we call it a strobili or strobilus for singular. They're usually at the tip of the stem. So you look toward the tip and you'll see this like tight cluster that looks very different than the rest of the body. And if you look very, very closely, you'll see that there is sporangia clustered in that region. And that's where all the spores are located. So they think this was the first stab at some kind of, it's like the forerunner to the flower in angiosperms. They think that this was like the first stab at how to kind of cluster together the spores into a larger overall structure, similar to a flower, is being used as a very special part of the angiosperm for sexual reproduction. So let's take a look at what I'm talking about here. So you're going to be trying to look for this on some of the seedless vascular plants. Now, not every singular seedless vascular plant has this, okay, but many do. So here on the left, you're looking at a club moss, and you can see how the strobili are at the tip, different shading of green. You can see how tightly packed together those leaves are. Right? And then over on the right, this is horsetail, and their strobili look a little different. They're a little bit more complicated in their structure, but again, it's that tight clustering of sporangia. And so if you crack those open, there'd be a bunch of spores in each one of those little tiny sacs of sporangia. Okay, so look for the strobili. Here's a, whoops. Here's an example of the strobili again. You can see the big difference in the locate, they look visually different from the rest of the body. Whoops, let me go on to this one. Here's another species of club moss. These are often referred to as ground pines because they look like little pine trees. And you'll often see them sold around Christmas time. They'll sell them as like little tiny Christmas trees. You might see that. So we have a lot of club mosses around here. So when you're out hiking around, look for that and look for the strobili. Then you'll also look at some spike mosses, which are in the same grouping as the club mosses. And a lot of those are for sale too. They're kind of distinctive and very pretty. And so you're going to look at one that's called the resurrection plant. So that one is fun because we're going to show you that it just looks all dried up in a ball, all brown, and it looks dead. And then we'll pop it into some water. And you'll see it within the time period of your lab. You should start seeing it activate. And so it has this amazing ability to go dormant for years and just curl up. And then that round shape helps it to roll around and with high wind, move around. And then when it gets into some water, it will activate. It's amazing. And then you can dry it out again and it curls back up, go dormant again, and then wet it again. So I've used the same resurrection plant for years to demonstrate this. It's amazing how hardy they are. Yeah, it's very impressive. So we'll look at those. Not exactly, no, the tumbleweeds are angiosperms, but it's the same strategy in the sense that it's a way for them to get into the right shape for wind, wind movement. So it's a really good strategy. But yeah, it's a different group of plants. We don't have any quillworts to show you, but that's another group we'll talk about. They're aquatic. They're often underwater in like shallow lakes and streams. You'll see them. So we'll look at those a little bit more carefully. And then I tried ordering some whisk ferns. Oh my gosh, they don't have them. The companies that I normally order plants from, for some reason they're having, I don't know what's going on, but they didn't have any whisk ferns. But we have some herbarium samples that we'll look at. Very kind of primitive looking, no true leaves. All right, so we'll look at some whisk ferns too and get a feel for them. And then with the horsetails we have around here. So if you go along Lake Superior bike path, often I can find them there. And then if you go anywhere where there's like rivers or ditches, that's like if the ditch stays moist for long stretches of time, you'll tend to see horsetails hanging out in like consistently damp areas. So look for that. But here you're seeing like two versions of the horsetail. So the one that is more bare and then has the strobilus on the tip. If you see it like that, and they refer to that as scouring rushes because the stem is ribbed and it contains silica. And so you can actually grab these and use them like if you're out camping and you burn your dinner. You can use these to scrub off the burnt pots and pans. And that's actually one of the heavy uses for them. And so you can see the ribbed pattern to the stem. And so that's there's a lot of silica in the cell wall. And then here's a close-up of the strobila. Very unique. And then this kind of weird looking structure there, you can see, and then down here, kind of the brownish gray. Those are actually the leaves, which are pretty useless in the sense of they don't do a whole lot of photosynthesis. It's the stem tissue that does it. And then some of the horsetail will look like that. This is all branches. That's not leaves. Those are just branches. And they call this horsetail because taken all together, it kind of looks like a horsetail. And so the two versions are scouring rushes and then the horsetails, but they're the same organism. They just have the distinctive look. So we'll have some of that to look at. And then the ferns are another huge group. So they're the largest group of the seedless vascular plants and probably what you're most familiar with when you think about this group. And so we'll look at some ferns. They're a little different now. They don't have strobili. They do something different. Let me just show you that. And then you'll look at this in lab. So where is it? So they form what's called sori, or here we're just looking at one, so it's a sorus. So if you've ever looked at a fern and you turn the leaves over and look at the underside, you might see the brownish kind of granular looking spots. And I remember the first time I saw those when I was little, I thought poor fern was diseased and I felt really bad for the fern plant, but it turns out you know that they've gone through sexual reproduction if they have that. So it's a good thing. So if you turn the leaf over and you see a bunch of brown dots, it's okay. It's a good thing. Okay, so those are sori, S-O-R-I, and that's how they do their, that's where they put all their sporangia and spores. They put it on the underside of their leaves. Okay, so they don't have strobili. They have to be do their own thing. So this is a microscope picture of a fern leaf, so that you're looking at the leaf on the top there, the fern frond. And then if you look at the bottom, so this whole entire thing, this whole cluster, we would say, oh, look at that sorus. And then each one you can see there's many, many sporangia. And then you can actually see the spores inside. Okay? So the sporangia, cluster of sporangia with spores inside, that entire thing they call a sorus. So ferns have got to be different. They got to do it their own way. So here's, again, this is what you have going on here. You can see some brown over here, a little bit of brown here and there. Sometimes they look green like this because they have a protective tissue over it to kind of just further protect and keep hydrated. And then here's a beautiful image under the microscope of a sorus with lots and lots of sporangia, lots of spores. Okay, so the ferns, you're going to see some different ferns as well from the greenhouse and herbarium samples. Okay, so you're going to get a survey of them in lab tomorrow. You'll look at a few microscope images as well of the seedless vascular plants. Okay, so we'll get you there tomorrow.